So yeah, you can see that I'm kind of picking up almost from the same slide as Greg was showing before. It has some subtle differences in there. So if I should kind of give a statement where I'm taking over, basically Greg laid a wonderful explanation groundwork in terms of what the data or model simulations are that we're working with. And we've reached the point where it's getting like kind of more complex. So I'm happy because I don't know too much about neurons, but when things get kind of complex and messy, then I'm happy because that's what I love working with. So when you look at this this raster plot here, you can start to see that, for example, here in this time region, we have a more or less synchronous population of neurons. All the spikes that we're seeing are occurring more or less at the same time and in synchrony. But as you're moving over to the right, you can see that the synchrony on the population level is pretty much lost. In accordance, your Z statistic is coming down from 1. So if you just look by this Z statistic, at this point you wouldn't necessarily infer that there is really synchrony going on or what really is happening in detail here. But if you start looking at the raster plot, there's still some structure happening in there. There's something local where things are actually coordinated. And the question is how you would get at this. Now, if you look at the title of the talk, it actually says functional clustering. So the main question about this region here that I'm going to try to answer in this talk and introduce methods about it is how to detect local clusters of synchrony inside of this population. I get to that later. I get to that later. Yeah. So also if there's mini questions, maybe ask them, but it's better to keep the questions to the end because the talk is recorded. So anyone who sees the recording will have a hard time with the questions during it. So, okay, so if I want to talk about clusters, the method I'll be introducing is hierarchical clustering. So I'm first going to talk about how hierarchical clustering in general works, and then how we take this to the functional clustering of these raster plots. So hierarchical clustering, I would sort of section this into 3 important steps. The first thing is if you have a population of points, samples, individuals, examples, you have to define a measure of distance between them. The next thing is that if you know the distances between the points, you have to calculate the linkage between the points and how they're connected. And lastly, as it's hierarchical clustering, it doesn't actually tell you how many clusters you have, but you have to find a cutoff point which will ultimately determine the number of clusters that comes out of that linkage structure. So that's the idea in writing, and I'm also going to show it graphically, how you would go through this sequence. So the first one is that you have to define a distance metric between the points. So obviously the squares represent points of an overall sample population, and what you would want to do is to go and define a somehow useful measure D that can relate the distance between two samples or points in this population, the points M and N. And with this, you can calculate the pairwise distance between each pair in this population. The second step is when you actually have this metric, based on the distances, you can calculate a linkage tree. So at the very bottom of the linkage tree, you would see so-called singleton clusters, or it's basically the data points by themselves. Each and every one are a cluster by themselves. Now you need an algorithm to construct this linkage tree. And what it does is to, in every step, take the, or at least in this version of the algorithm, it takes the 2 clusters that at this very iteration of the algorithm are the closest together in terms of distance and links them into a new cluster. So with every step, you arrive at a new state where you have 1 cluster less because you have combined 2 clusters into 1. And if you execute this to the very end, you will realize that you're actually ending up with just 1 cluster. And that's the very top of this tree. Now, as you have this linkage tree, it doesn't really tell you yet how your clustering finally should work out because you still have lots of choices in terms of how many clusters you got. So maybe the most difficult question in all of this is to determine the cutoff point for this tree. So I think it kind of works on the projector here too. You have to know where to cut, where to put your scissors. And that's a key question because it will determine the number of clusters that you end up with and ultimately the structure that you're extracting from your results. But so much for the general idea of hierarchical clustering. And now we're going to take this over to actual functional clustering, or the example that we're working with here. And in showing this, I will show the definition of the distance, again, the linkage tree that comes out, and also how we actually determine the cutoff using the definition of modularity. And in doing so, I'm actually really heavily drawing on this paper from Susan Feld. And if you want to see where these methods are coming from, I would really recommend checking this paper out. And if you're interested in seeing even much more complicated methods than this, then that's actually the same paper because they go way beyond this too. I didn't. Okay, so here is a small section of the raster plot that we were looking at in the very beginning. And let's say we have a neuron M, and we have a neuron N, and we want to compare, or we want to calculate what actually their distance in terms of the spike trains is, in terms of their synchrony between the two spike trains. For this, what we would use would be the average minimum distance between the two spike trains. So you calculate this in the two directions, from M to N, but also from N to M. But let's, for now, look at the one that calculates from M to N, so just the top one. So for every spike that occurs in the trace of the neuron M here, you would look what is the very closest spike time in the trace of the neuron N, and you calculate for this pair of spike times the distance or the difference between these spike times, and then you basically do an average for all the spike times in the trace M. And the average of these spike time differences tells you the distance coming from these two traces. Then you do it vice versa and basically add them up, and that's your average minimum distance between the spike trains. And with this, we have defined a distance between them, and we can actually calculate how far apart they are. Based on this distance, we can lay out this distance matrix here. So you can see wherever things are colored in a dark scale of gray, things are not very far apart in terms of the spike timing. And you can already kind of guess the cluster structure that would come out of here. So wherever you see these dark squares, things are connected very closely. Where it's very bright or light gray, it actually means there's a high functional distance between the neurons, so they're probably not going to be together in a cluster. Based on this matrix, you can actually feed this into the algorithm to calculate the linkage tree. So you would end up here. And what you see in terms of the y-coordinate as you go from the top to the, or actually you should start from the bottom. If you go from the top to the bottom, the y-coordinate is the linkage difference. So it's basically by joining the two clusters, you basically take the distance between the two clusters that are being joined at this moment. So you would, for example, look for a point where there's a huge linkage difference because you would have two clusters that are very far apart and they're being joined. So this would be sort of a suggestive point for cutting this tree. And that's what's often is done if you just have one sample that you're looking at. You make this linkage tree, you look at what's a point that looks promising and you just cut it off and you go with that. I think it's often also accepted. It was accepted for me. But if you have lots of simulations, you might want to have an automated way of doing this that's also a little bit more consistent or systematic. And that would basically be the modularity criterion that we use here for replacing the cutoff. So you can see that we have the cutoff put in here, and it's determined from the maximum in modularity as you go through the different possible numbers of clusters in here. So I quickly wanted to give an intuitive idea of what modularity actually means. modularity goes from a value of -1/2 up to +1. And if you calculate the modularity and you would find a value of 0, it would mean that you have two groups. So here given in, it's visible, in blue and in cyan. But there's basically no ordering of these groups, but there's also no ordering in the population per se. So your classification into clusters is entirely mixed up, but at the same time, there's also no structure in your population. A different case could be that you actually have a structure in your population. So you can see that on the right here. You have groups that are clearly separate in your population, but also your clustering classification is very successful at telling the groups apart. So your sorting into classes also leads to a high separation in the population. So here you have a high value of modularity. The opposite case where it turns negative is that you have a population that actually has a structure in itself. You do a classification and it just comes out basically random. So your two classes have nothing to do with the actual structure inside of your network or sample. And that's a negative modularity. So what you want to do in terms of having a successful detection of the clusters actually present in your sample is to find where your modularity is the highest. And that is what here this graph on the right does. As we go and we start from the top, where we just have one cluster at this point, corresponds to the very left in this graph. And now as you move the threshold for cutting it off down and down and down here, you'll be going to the right. And at some point you hit a maximum for the modularity. So that looks like the best clustering that you can apply to this population. And that's the one that we would pick from here. So that answers the question where to put this cutoff point, how many clusters to take from your linkage tree. Oh yeah, here comes the tribute to Fred. He asked me, so how do you do this in MATLAB? One part is you actually have to go to this paper, understand how to do a couple of computations, like the distance, for example. But mostly for the clustering itself, there's a fairly standardized toolbox for this. Pairwise distance, we can't use it here because we calculated the distance in our own special way. The linkage structure can be calculated with the command linkage. You can visualize the tree using the command dendrogram. And then finally, if you know the number of clusters that you want to have, you use the command cluster, give it an argument that sets the number of clusters, and it's going to tell you the groupings that come from your tree. Yeah, that are the commands for MATLAB. And yeah, I also wanted to show some example results. So here, again, we go back to that raster plot. But to also answer Fred's question from the beginning, so what actually is the ordering of the neurons that we see, say, from 0 or 1 to 150? Is there anything giving them structure? And the talk started with Greg talking about spatial relationships between the neurons. So you can see here on the left that we have created a sort of elongate field of neurons that are spaced in a two-dimensional domain. And you can also see where the clusters are actually showing up in this field. That's the, well, they're not circles, they're actually convex hulls, but they're describing where the clusters are contained within. And you can see that here for the conditions where we have this local clustering, they're actually really localized clusters that contain a segment of this field, and certain other segments fall into different modes of synchrony that are independent of the other parts of this field here. So that's the idea, that we could arrive at actually finding these spatial clusters within this field and see where the synchrony is located in these neurons. Does this explain like how they spatially order? So basically, if you go from in the y direction from the lowest one that would be mapped to here, it increases in the y direction, so they're y sorted, and 150 would be the topmost one here. Yeah. So this is one example. As we change a model parameter, we can actually see how statistics for the clusters are changing. So for 3 different model parameters, R, which is the distancing between the neurons on average, you can see that there are different things happening in this population now. So for example, you would see that you have, for a small r, the circles up here, there is an increase of the firing rate inside of the clusters, and the firing rate in the cluster also depends on the cluster size. So a bigger cluster seems to be firing at a higher rate, smaller one at a lower rate. But this effect is also dependent on the model parameters. So if you have neurons that are on average further apart, you see that the cluster size does not actually really have an effect on the firing rate. Another one you could look at is the cluster span. So if you have a cluster, if it would span the whole field of neurons, it would have a span, it would basically have the maximal span here, which is around 40, 45. So it would mean that the cluster goes over this whole field. And if it has one that is lower, it means it only has a segment. So you can see that for the low R value, with increasing cluster size, so more neurons being in one cluster also means that the cluster is bigger. But this relationship is lost for the high R values, so for different versions of the model. You can see that basically even if a cluster would have 10 neurons, it can already span all over the whole field of neurons. So it would be looking kind of like this. On the left hand, you have the locally synchronizing clusters for the low R value. And they're really nicely separate, as you can see here. Then for the intermediate R value, you can see that, yeah, you get a cluster. And this cluster is basically the whole field of neurons. So everything is synchronized. That's the case that Greg was talking about. And then as we go to an even higher R value, you see, well, we get a bunch of clusters. But these clusters are not actually localized clusters, but they're basically kind of random associations, like random synchronous neurons that occur. And yeah, with this picture, I would go back to where we started and would ask if there are questions to me or to Greg as well, because I asked him to save the questions for now. Thanks. Yeah, oh, what's the size of the cell?
Oh, I mean, in the model, the cells are points. So
right.
The models are size
yeah.
Yeah. Yeah, we're sort of taking the distance here as like the distance between. Yeah, or like the distance between the cell bodies. So, you know, our two cell bodies touching each other. Yeah, that's right. Yeah, I mean the narrative of the talk was that was totally inappropriate for the question. Really, in the paper we worked it in, and they're sort of generalizing some of the results from the all caps neuron to this. But yeah, as far as the talk goes, no, we didn't do that. That's right. Really, I mean, the oscillator model, you know, is such an abstract model of neurons that you can really kind of take it wherever you want. For us, we use the coupling as an aphaptic coupling. So you see, like, if you have two cells next to each other and one fires an action potential, you see this little spikelet in the membrane voltage of the other one, where there's this little, you know, transient fluctuation of, you know, 0.1 millivolts, you know, something on the order of microvolts. And so it's that kind of coupling that we're trying to capture with the oscillator model. Yeah, that's right. Yeah, that's exactly right. Thanks.
So, no, it's fairly simple. It's an M by 3 matrix. So M is like clusters minus 1. It's fairly simple to work with, and it's basically a workflow. Like if you have just points in a 2-dimensional space, 3, and you can use a Euclidean distance, you throw the point coordinates into pairwise distance. The output of pairwise distance goes into linkage. The output of linkage goes into cluster. You tell it the cluster number, and you're done. Yeah. Yeah. The only difference here is that instead of the pairwise distance, which is predefined, we use the functional distance. And we use the trick of modularity to find the cutoff point, which usually you would just do by eye or some other method. Yeah, that's it.